Okay, now we will discuss on anatomical features in elucidating taxonomic affinities between different plant groups. So, anatomical features have played an increasingly important role in elucidation of phylogenetic relationships. So, anatomical characteristics are investigated with the help of a light microscope, whereas the ultrastructure, so the finer details of contents, and micromorphology, so the finer details of surface features, are brought out using an electron microscope. We will discuss this in the next videos, inshallah. So, anatomical work of taxonomic significance, especially in the context of evolution, especially to determine the primitive and modern angiosperms. That's one of the example. Okay, first talk about the wood anatomy. So we talk about wood anatomy. Wood represents the secondary xylem constituting the bulk of trees and shrubs. They can form through the activity of vascular cambium. It primarily consists of tracheids and vessels. So so investigation. So we're talking about wood anatomy. We talk about the find something that we can the anatomy that we can the structure that we used we looked through the the microscope, the optical microscope. So investigation of these two wood anatomy, which is the tracheids and the vessels, need to use the microscope for the fine detail examination. For instance, so vessels are absent in gymnosperms. Okay, but it is commonly believed that there has been a progressive evolution of angiosperms from tracheids, okay, to long narrow vessels elements with slanted scleriform perforation plates, to short broad vessel elements with simple perforation plates. So these are the changes between the gymnosperm to angiosperm. So the demonstration of vesselless angiosperms, for instance, in the Winteraceae, in the Trochodendraceae, also having other primitive features, so has led to the conclusion that angiosperm ancestors were vesselless. So nodal anatomy, nodal anatomy here has considerable significance in angiosperms systematics. The number of vascular traces entering leaf base and associated gaps left in the vascular cylinder of stem at each node are distinctive for several groups. So the node itself may have singular gap we call as unilacuna from single leaf trace or three leaf traces, or which means two additional commonly entering stipules, or three gaps, sorry, like a we call it as a trilacuna. So these are can be associated with three leaf traces. So like in this figure. So this one unilacuna. So and then this is the trilacuna. Okay. Uh, so the genus Elysium has been separated from Winteraceae because of these unilacuna nodes. So there's continuous pseudosiphonostil and the absence of the granular material in stomatal depressions. Sorry for my uh, Greek influency. Okay. Again, that's for wood anatomy. Also, when you talk about some of the anatomy, one of the important anatomy of the wood is is the trichomes. Okay, trichome constitute is the appendages of epidermis, which may be glandular or glandular. So we can differentiate many plant families based on this character. So example, the Brassicaceae, Lauraceae, and Moraceae have non-glandular trichomes. So maybe in the forms of simple unicellular. So A, yeah, is unicellular and multicellular. The B here. So unicellular and multicellular. So atriplex, here, the atriplex here. Okay, uh, has a bladder-like unicellular glandular hairs with few celled stalks. Okay, they have stalk here, and basal cell and the and also they can secrete salt. So this is a special character for halophyte like this plant. So some trichomes also can secrete nectar, like in Abutilon, Rheum, and Rumex. So the stinging hairs, okay, like this, like is in this stinging nettle in the Utica dioica, okay, is highly specialized with silica tip, which readily breaks when hair is touched. Okay, so the broken tip is sharp like a syringe and easily penetrates the skin, injecting irritating cell contents. So therefore, trichomes hold considerable promise in systematics of angiosperms. Trichome has been considerable help in cruciferae, the order of cruciferae, especially in the genera Arabis and Arabidopsis. Trichome characters also are very useful in the large genus of Astragalus. So like in the Himalayan species, the Hedera is distinguished from its European relative, so Hedera helix, in having scaly trichomes as against stellate in the Hedera helix. That's for trichomes. So we can talk about epidermal features. So these epidermal features are also of considerable taxonomic interest. I'll try to discuss further on SEM epidermal features under ultrastructure and micromorphology later, inshallah. These stomatal types here, different stomatal types here are distinctive of certain families, like in the family of Ranunculaceae, which have the anomoctic stomata. In the Brassicaceae, we have anisocytic. In the Cryophyllaceae, they have a diacetic. In the Rubiaceae has a parasitic stomata, and finally the Poaceae has is more on gramineous stomata. So closely related families like Acanthaceae and Scrofulariaceae can be distinguished by the presence of diacetic stomata and anomocytic stomata. So like the Acanthaceae is diacetic, and Scrofulariaceae is the anomocytic stomata. But all the stomatal features, okay, however, are not always reliable. For instance, in the Streptocarpus, the cotyledon have anomocytic, while the mature organs have anisocytic stomata. So it depends on the species that you work on. So you need to be very careful on looking on these characteristics. So not much on leaf anatomy as compared to its morphology, but leaf anatomy. We talk about leaf anatomy. We talk about more on leaf morphology instead of leaf anatomy. But leaf anatomy has been special taxonomic help in certain family. The occurrence of the C4 pathway. This is the especially in the association with the Kranz anatomy. What we call as Kranz anatomy is a dense, thick wall chloroenchymatous bundle sheath and the mesophyll and the simple mesophyll. So this has resulted in revised classification of several genera of grasses. So the Kranz anatomy is very crucial. So between the C4 and C3 pathway. So these are one of the example of how how the taxonomies use the leaf anatomy to differentiate, especially in the grasses family. Leaf anatomy. So finally, floral anatomy has been one of the thoroughly explored areas with significant contribution to the understanding of the phylogeny of angiosperms. The main study on floral anatomy was based on the vascular traces in the carpels. So various genera of the family, like in the Ranunculaceae, have confirmed, okay, this is Ranunculus, have confirmed the origin of the akin in the Ranunculus from the Delphinium. So Ranunculus has the akin and the delphinium here has the follicle. So they can differentiate between the two. So this is based on the successive reduction in number of ovules. So for ultimately to one, so they can differentiate based on the number of the ovules there. So in the gonophyll theory, the study of vasculature of carpel and other floral parts through the clearing technique. This theory believe uh, that the angiosperm carpel to be a modified dichotomous fertile branch adnate to the petiole of a leaf. So, and then when talk about the inferior ovary, in angiosperm has been formed in two ways, either in the appendicular origin, where it's formed by fusion of calyx, corolla, and their traces to the ovary wall, and in this case, all vascular traces have normal orientation, i.e., the phloem towards the outside, or by axial invagination, so formed by depression of the thalamus. The inner vascular traces have reverse orientation, i.e., the phloem towards the insides. Okay, and then there's, excuse me. Of course, the studies on floral anatomy have confirmed that in large that in a large majority of families, they have been used to differentiate. I can say the majority of families in the plant family, and of course the inferior ovary is of the appendicular origin. Only in a few cases like in Rosa and Cactaceae, the origin by axial invagination of the thalamus. So again, anatomical characteristics are investigated with the help of light microscope. Anything that you use microscope to analyze the anatomical features in this we can use we can characterize in this what we have been discussed in this lecture. If you want to go more details beyond the light microscope, you can use electron microscope. For more finer details of contents in ultrastructure, you can use the transmission electron microscope, and the finer detail of surface features in micromorphology, you can use the scanning electron microscope.